So just to reiterate where we've been covering population genetics, and we've been talking about microevolution. When I talk about microevolution, I'm really talking about shorter term changes in allelic and genotypic frequencies within a population, like from generation to generation. So this is the sort of stuff that we were calculating when we were looking at Hardy-Weinberg equilibrium. And we talked about some of the agents of microevolution. So what are the things that cause allele frequencies to change in a population, or genotypic frequencies to change in a population? And those would be natural selection, genetic drift, gene flow, mutation, non-random mating. So these are all the things that cause a population to evolve by changing the allele frequencies in a population. So today we're going to talk about macroevolution and speciation. So when I talk about macroevolution, what I'm talking about is larger scale evolution that happens over sort of larger and longer periods of time. So these microevolutionary processes add up to macroevolutionary processes in speciation, or basically the creation of new and different species. And so if you think back to that first slide that we looked at on the first day of lecture, where we talked about, the 1.8 million described species that are out there and the huge diversity of organisms that we see on planet Earth, from bacteria all the way to sort of more complex organisms like the proboscis nose monkey and the goblin shark that we're looking at here, and all these other organisms that we're looking at. This, of course, gives rise to the question of where did all of these organisms come from? And how did we go from these very sort of single-celled basic organisms to these much more complex organisms that we're looking at here? And the answer to this question is this process of speciation. And what speciation is a splitting event that creates two or more distinct species from a single ancestral group. So you start with an ancestral group. That ancestral group eventually over time, so we're talking about hundreds, thousands, hundreds of thousands, millions of generations, potentially millions and billions of years, these ancestors can split into different species. So to understand this process of speciation, we have to understand what a species is in the first place. So hopefully this is not the first time that you guys have looked at this. I guess the figure isn't the right word for it, but this is a breakdown of all the major sort of taxonomic groups of organisms, their taxonomic hierarchy of organisms, from domain all the way down to species. And so species is the most sort of specific taxonomic group that you can get into. And so if you think about a dog, the species name for a dog is Canis lupus, where Canis is the name of the genus and lupus is the name of the species taxonomic level. But whenever you describe the species, you call it Canis lupus. So you call it by, it's what's called the Latin binomial, or the species name, that includes the genus name and species name. So this would be Canis lupus. And just a sort of smaller, finer point is when we're talking about species, the individual, or the sort of, the plural of species is species. And if you're talking about one individual, it's not a species, it's an individual species. So the singular is also species. So the big question we want to address today, or one of the big questions we want to address today, is how do we identify an individual species? So for example, when you look at this poodle here and this tiny little dog here, these are both considered the same species, Canis lupus, but they look obviously very, very different. So how is it that two individuals that look so different can be called the same species? Whereas over here on the left side, we're looking at these two different species of trogons that to the untrained eye look pretty much exactly similar, except for some of the banding patterns on the tails maybe. So the greenback trogon and the gartered trogon, these are considered two species. So how is it that something this similar can be considered different species and something that looks this different can be considered the same species? And the answer to that is all about reproductive isolation. So when we talk about a species, what we're talking about are groups of actually or potentially interbreeding populations that are reproductively isolated from other such groups. So essentially what you're talking about when you're talking about reproductive isolation is you just can't produce viable offspring. So as long as two individuals can potentially produce viable offspring, And when we're talking about viable offspring, we mean offspring that themselves are capable of going on to reproduce. Those two individuals are not, or are considered the same species. If you cannot produce viable offspring, then you're considered different species. So for example, in theory, you could take this poodle and this little schnauzer dog or whatever it is. They're not reproductively isolated, and they could produce viable offspring that can go on to reproduce itself. So for that reason, these are not considered different species. Whereas these two trogons, they're incapable of producing offspring. They can't reproduce, they can't mate, so they're considered two different species. They are reproductively isolated. So how are different species made? So how do we get two different species that are reproductively isolated from one another in the first place? One of the major processes that gives rise to this is something called allopatric speciation. And this is a three-step process that involves separation of populations, over time the genetic divergence of those populations, until eventually those populations become so different that they're reproductively isolated from one another, in which case they're considered different species. So this is speciation. So let's look at this process in a little bit more detail. The first step being separation of populations. So you can imagine a parent population that you're looking at up here in the gray lines that somehow gets split into two populations. So maybe a river forms between these two populations, maybe over millions of years, a mountain range rises between these two populations, but it splits it up into population A and B. And see, we're moving here forward in time. So the populations are first separated. Now that they're separated, we begin to see genetic divergence in the populations through those microevolutionary processes that we talked about. So you're getting changes in the genes or allele frequencies in population A versus B through mutation, natural selection, genetic drift, non-random mating, any of those processes that we talked about, such that these populations begin to become more different from each other, genetically speaking. So these populations are what's called diverging. They're genetically diverging from one another because they're under potentially different selective pressures. So different mutations are arising in the two populations, different types of drift are happening in the two populations such that they're becoming more and more different, until eventually those populations become so genetically different from one another that the populations can no longer successfully interbreed. So if I take an individual from this population, an individual from that population, put them together to try to get them to breed, it doesn't work. They're reproductively isolated because they're so genetically different from one another. And now they're considered to be different species. So if we look at this idea of separation of populations, I want to talk a little bit more about the actual processes that can cause a population to separate. The first sort of mechanism for separation is just dispersal and colonization, where one individual from a population arrives at a new isolated place like an island or something that's geographically isolated from the parent population. So this is one mechanism of separation. The other is something called vicariance. So vicariance is basically a change in the geographic habitat that the population's in that splits the population in half. And this sort of example here is basically a river gets diverted such that it splits the two populations in half so that they're no longer able to interbreed with one another. So this could happen this way. As I said before, you could have mountains uprising and splitting the population over millions of years. You could have a lake full of fish that the lake dries up such that a land border forms between it and it turns into two lakes that separates the population. There's plenty of different ways that a vicariance event can happen, but the key thing is that the population gets split in half. So you have two newly isolated populations that are no longer able to mate with one another, basically just due to some geographic barrier. So why is this critical for this speciation process? Why is it so important that these populations cannot mate with each other? Go ahead and just type that into the chat if you can. Why is it so important that these populations are physically separated? So I'm getting a couple different answers right now. And the key thing here, so you've got these two populations that are separated from another. They cannot interbreed. And so what this stops is gene flow between the two populations. So remember, in this process of allopatric speciation, you've got two populations getting separated. And potentially those populations can very early on, when they're becoming genetically different from one another, so they have different sort of selective pressures or different mutations arising in those populations, they can become different. But if they're interbreeding with one another, any of those differences that arise in those populations get shared across them. And so it counteracts the accumulation of genetic differences. And so you need this physical separation to stop gene flow between them, so that those populations can become more and more genetically different from one another. If you have them interbreeding, they're going to share alleles and genes, and they're going to become similar to each other in terms of their genetic makeup in the populations. So it's really important to prevent gene flow for this allopatric speciation process to happen. So just to sort of drive that home, the second step, the divergence, or the genetic divergence of the populations, is this key step. You need the separated populations to evolve independently from one another for them to become genetically different from one another, such that any change that happens in one population, in the allelic frequencies or the genotypic frequencies of one population, doesn't occur in the other population. So they're becoming genetically different in terms of their allele frequencies, the alleles that are there, and the genotypic frequencies that are in the two populations, such that they diverge over time. So just to sort of give you an example, you could have this population get split in half down here, where you've got the red individuals on this side, where they have a selective pressure of a hawk that just doesn't like red salamanders for whatever reason. And so you have strong selection for red individuals here, such that you have a big change over time in the allelic frequencies in that population, along with other changes that are associated with this sort of selective pressure. Whereas here on this side of the population, you've got a green snake that has a hard time just seeing green individuals in this habitat or whatever it is. And this is the major selective pressure. And so you have the divergence of the populations over time, such that they eventually become so different they can no longer interbreed. So let's look at a case study of allopatric speciation. Let me pull up this video real quick.
Meet a type of terrestrial salamander found in California. Now meet another, and another. These salamanders are members of the same species, so why do they look and behave so differently?
You guys are seeing this, right?
In California, populations of these salamanders became separated geographically, evolving over millions of years to adapt to different environments. They've changed so much that they are on their way to becoming entirely separate species. David Wake of the University of California, Berkeley explains.
The history of this species complex began about 10 million years ago in the ancient redwood forests of Northern California, where we have today this form picta, which is highly variable both genetically and in terms of coloration. There were two migratory routes followed by animals as they moved into Southern California. One along the Sierra Nevada mountain chain, where animals moved into the forest region. The other along the coastal mountains. They avoided the Great Central Valley.
The populations that gradually moved down the forested region of California relied on blending in with their environment. Animals with good camouflage tended to survive more and produce offspring with similar traits. As the salamanders progressed south over millions of years, the markings became more distinct.
At the end of the chain in Southern California, we have this form clouberi, which is the extreme of variation, showing the largest blotches, most bold marking. These were all then camouflaged organisms that were hiding from predators. Along the coast, a different strategy was followed. Here, the organisms adopted the color pattern and behavior of dangerously poisonous newts. And they became, instead of camouflage, they were advertising their resemblance to these dangerously poisonous animals and gaining an advantage because of their mimicry. So what happened is that you had two differently adapted lineages moving to the south, and by the time they reached the southern end, they had essentially evolved into different species. But at certain areas, for example, in the Palomar Mountain area in San Diego County, we get these animals, which are hybrids, and the hybrids are neither similar to the dangerously poisonous animals, they're not good mimics, nor are they good animals in terms of camouflage. So these organisms are essentially dead ends.
These hybrid offspring are not well adapted to their environment and are therefore less likely to survive. The two parent subspecies that produce the hybrids are well on their way to becoming entirely separate species, evolving just as Darwin described in his theory of evolution.
So this is one example of how these microevolutionary processes can add up to macroevolution or speciation, where you've got different selective pressures causing these different species of salamanders to slowly diverge from one another to the point that they become different species. So in any of these cases, something called secondary contact can occur. So secondary contact is just after a population has gotten split apart, it comes back together. So for example, you've got this fish population that's split in half, and over millions of generations, two very different sort of populations, potentially different species, have emerged. And then after a couple decades of big rains, those populations come back together. So the big question is, now that these populations have diverged genetically, can these two different populations or individuals from these two different populations interbreed? Are they reproductively isolated or not? So is gene flow going to resume and sort of counteract the speciation process? Or are they going to be reproductively isolated, in which case we can consider these two different species? And so there's a couple different outcomes of this secondary contact. The first being hybridization. So this is just a sort of silly example, but you can imagine, these different species of rabbits come from different populations and they're different from one another, but they are still capable of interbreeding. They can hybridize or produce a hybrid offspring that's an individual from two different populations, or an individual from parents from two different populations. And there's two major outcomes that you can have from hybridization. You can either get viable offspring that are pretty much equal in fitness to the parents, so they're not selected against. Or you can have viable offspring that do have some sort of fitness cost, so they're not as fit as the parents, and therefore they're selected against. When I say viable, what I mean is those offspring themselves are capable of reproducing and passing their genes on to subsequent generations. And so if you have viable offspring that are not selected against, that are not any less fit than their parents, this is essentially equating to normal amounts of gene flow, and this should slow or counteract the speciation process because it essentially encourages hybridization. It encourages gene flow between those two separated populations. And so it's going to counteract the speciation process. Whereas if the offspring are not as viable or they're selected against, this should actually reinforce speciation. Okay, so the example would be what we just heard about, where you have these two different sort of subspecies of salamanders, Ensatina salamanders, and they'll crossbreed here with the sort of spotted camouflaged individual crossbreeding with this individual that's made to mimic poisonous salamanders or toxic salamanders. And so these two will interbreed to make this hybrid individual that's really not blending in really well because it doesn't have a good camouflage pattern, and it's not making this sort of warning signal that I'm a toxic individual to predators. And so it's not ideal for either habitat. And so those hybrids will be selected against, and this will lead to something called reinforcement. All that reinforcement is just reinforcing speciation by selecting against hybridization. So those parents that choose for whatever reason to hybridize their offspring aren't going to survive as well. And so those parents will be selected against. Whereas those individuals that don't hybridize for whatever reason, they're going to be selected for because their offspring are going to be more successful in subsequent generations. So other potential outcomes of this secondary contact is you can't hybridize. So parents come together and they are reproductively isolated. They can't hybridize. So once again, this is a very sort of silly example, but you can imagine a rabbit and a chicken trying to produce offspring. Obviously, they're different species. Obviously, they cannot produce viable offspring. They're reproductively isolated. And there's actually two different types of reproductive isolation. The first one being something called prezygotic reproductive isolation. So remember, a zygote is a fertilized egg. So a sperm fertilizes an egg and forms a zygote. So prezygotic isolation basically stops any sort of fertilization from happening. It stops sperm from meeting egg. So that is a type of prezygotic isolation. And this is equivalent to speciation. So if two species are prezygotically isolated, they are considered to be different species because they are, by definition, reproductively isolated from one another. So there's different types of prezygotic isolation. One is ecological. So this is basically just two different species occur in the same area, but breed in different habitats. So for example, you have Iris fulva that only grows in creeks, and you have Iris hexagonia that only breeds in marshes. Because they're in separate habitats, they won't cross-pollinate with one another. They are prezygotically isolated. They won't fertilize one another. Other types of prezygotic isolation, temporal. So this is just a difference in timing of breeding. An example of this would be Bufo fowleri and Bufo americanus, two different species of toads. One reproduces in late spring, the other produces in early spring. They don't cross over so that they're incapable of fertilizing one another. So prezygotic isolation. One can't fertilize the other because they're not breeding at the same times of year. You can also have behavioral isolation, prezygotic isolation. So behavioral is just some behavior that stops two different species from interbreeding. One great example of this is fireflies. You may not know there's actually tons of different species of fireflies. And many of these fireflies differ in their courtship behavior, specifically the lighting patterns that they have. And so what you're looking at here are a list of different names of species of fireflies. And here you're looking at the flashing patterns that they do on their butts to attract mates. So males will flash in a particular way for females to come and find them. And they have very specific flashing patterns that are specific to each species so that a female knows it's an individual of the same species and she'll go to that individual. And so you have a behavioral courtship behavior in this case that's species specific, such that one species only attracts the same species and not others. The last type of prezygotic isolation I want to talk about is mechanical, where either genital or morphological features are incompatible across species. So snakes are a great example of this. Snakes have these things called hemipenes. They actually have, in this case, males have two penises. And the sort of formation and the shape and the barbs that are on those penises are species specific, such that individuals of one male snake can only breed with, physically speaking, females of the same species. And in the same sense, the females, cloacas, so that's sort of like a snake vagina, are only receptive to penises of those males of the same species. And here you can see different shapes and sort of barbing patterns on snake hemipenes. So this is an example of a mechanical prezygotic isolation. Okay, poll question. And D is the correct answer. So a spermatophore is essentially a little package of sperm that oftentimes are species specific in the sense that the male's spermatophore has a very specific shape to be receptive, or females are only receptive to those specific shapes of the males of the same species. So for example, you can see right here, here's the shape of a newt spermatophore, and it has this very sort of crazy ornate shape that's specific to female newts of the same species. Okay, so that's prezygotic isolation. You can also have something called postzygotic isolation. So postzygotic isolation is just after the formation of a zygote, you essentially don't have viable offspring. So you can have fertilization occur and the offspring may develop, but they're not viable in the sense that they're incapable of reproduction. And this will once again reinforce speciation. So types of postzygotic isolation, you can basically have hybrid inviability, where even though you have a fertilized egg, that egg, when it goes on to develop, it basically is aborted. So it won't make it the whole way to sort of birth. You can also have hybrid infertility. So the classic example of this are mules, where you have a mare and a donkey interbreed. These are two different species. When they interbreed, they make a mule, but that mule is sterile, so it's incapable of reproducing itself. In both of these cases, hybridization or hybrids are going to be strongly selected against. They're an evolutionary dead end. They're not going to pass on genes to subsequent generations. And in any time that you have these postzygotic isolation mechanisms, these should lead to reinforcement, okay? And what I mean by that is you're going to select against individuals that hybridize, strongly select against individuals that hybridize, and strongly select for prezygotic isolating mechanisms. So any parents that hybridize are going to have sterile offspring. They're going to be strongly selected against, and there should actually be selection for prezygotic isolating mechanisms in those parents. And so European pied and flycatchers are a great example of this sort of reinforcement and selection for prezygotic isolating mechanisms, where you can see here, this is the sort of distribution of the collared flycatcher in blue. This is the distribution of the pied flycatcher in green. And the yellow area represents the hybridization zone. So this is the spot where those two distributions overlap, where the pied flycatcher overlaps with the distribution of the collared flycatcher. And you can see that in the area where only the collared flycatcher is, it's sort of dark colored with a light belly, and the pied flycatcher is very similar colored with a dark color and a white belly. But in the hybrid zone where they overlap, we see the pied flycatcher has this much lighter brown color, and that's basically a signal showing I'm a pied flycatcher, you don't want to breed with me, you don't want to hybridize with me. And so we have strong selection here for a prezygotic isolating mechanism in terms of just the physical color of the organism so that they don't accidentally hybridize with one another. So this is an example of reinforcement and selection for prezygotic isolating mechanisms to stop hybridization. Okay, another clicker question. C is the correct answer. Geographic separation, divergence, reproductive isolation. So allopatric speciation is one process, and probably the most common process that leads to speciation. But there is another process called sympatric speciation, where populations are not geographically separated or isolated from one another. They can also give rise to new species. So sympatric speciation is just speciation without geographic isolation. That's the definition of sympatric speciation. And there's two major processes or sort of types of sympatric speciation. One is sympatric speciation can occur through a process of disruptive selection, or it can happen through polyploidy. And we're not going to talk about polyploidy, but we will talk about how disruptive selection can lead to sympatric speciation. So when we're talking about sympatric speciation, all that it is speciation when there's potential gene flow between the two diverging populations. So in this case, you have an ancestral population, and in that ancestral population, there's no physical separation between it. Individuals in this population can potentially interbreed with one another. But disruptive selection occurs, and eventually what you end up getting are two genetically distinct subpopulations that eventually become so different that they diverge substantially enough that they're two different species now. They can't interbreed, they become reproductively isolated. And soapberry bugs are a great example of sympatric speciation. These are bugs that are in the southwestern or southeastern US rather. And they feed on these very specific types of plants in the Sapondaceae family. And the fruits of those plants, basically the way they feed on them is they stab these sort of needle-like mouth parts into the fruits of these plants. And what happened is in the mid-20th century, these non-native fruits got introduced into the soapberry pug populations. And those the non-native fruits had a very different, smaller shape. And as it turns out, soapberry bugs, their sort of mouth part or beak length corresponds to the size of the fruit that they feed on, such that individuals that feed on smaller fruits or specialize on smaller fruits are going to have shorter beaks. Those that specialize on larger fruits are going to have larger beaks. And what you saw, or what was observed in the populations over time, is disruptive selection. So you had selective selection for individuals with smaller beaks to feed on those smaller fruits, and larger beaks to feed on those larger fruits. And as these bugs became more specialized on the particular fruit sizes, they tended to stay and mate near those host plants that had those particular fruit sizes, such that there was reduced gene flow between populations of plants on one, or populations of insects on one plant versus the other plant species. Those unique mutations started to accumulate over time until eventually reproductive isolation may evolve in these populations. So they haven't officially split into different species yet, but it looks like they're going that way because they become so specialized on those two different fruit sizes. So the take home here is that you've got disruptive selection for two different sort of extremes of phenotypes, which is leading to genetic divergence in the two subpopulations. And this is ultimately going to result in them becoming so different that they become reproductively isolated and become different species. So just to sort of summarize what we've talked about here, I want to point you to this figure in your book, figure 21.16. And the main sort of take home here is that all of these microevolutionary processes, natural selection, drift, mutation, non-random mating, can all contribute to genetic differences emerging in populations that eventually lead to species becoming so different that they're reproductively isolated and are different species. And what you're looking at in this figure is just that sort of process happening through time. So here we're in the past in a parent population, and then we're moving forward in time to the present in two different populations, population A and population B, that get split in half because of a vicariance event where this sort of river gets split in half. And in population B, you have these blue mutations, which represent neutral mutations that arise through genetic drift to become eventually fixed in the population, where 100% of the population has those alleles. And so here, genetic drift is changing the allele frequencies in this population, whereas in this population B, you have natural selection occurring, where this red individual represents a new mutation that gives rise to a larger mass. Natural selection is selecting for that such that that becomes fixed in this population. And so here we have genetic drift changing allele frequencies in this population. Here we have natural selection changing genetic and allelic frequencies in this population such that these two populations are diverging.